Human-Induced Pluripotent Stem Cells Initiative, HipSci, is generating a large, well-characterized, high-quality reference panel of human-induced pluripotent stem cell lines for the research community. These lines are created from tissue donations from both healthy and rare disease volunteers. This video and its associated document demonstrate how to navigate through our cell line browser and purchase a line of interest. Working with clinicians from across the UK, the HipSci project has successfully obtained tissue samples from volunteers across 16 cohorts. Our largest cohort by far is our healthy volunteers. We believe they provide a powerful resource to study common variation within the human population. This then helps to better understand disease. These are the additional 15 rare disease cohorts in our collection. The successfully derived iPSC lines from all 16 cohorts are or will be available to the scientific community via two cell banks, ECAC and eBisc. Details of all the cell lines HipSci derives are collated in our Cell Lines and Data browser. The easiest way to launch our Cell Lines and Data browser is via the push buttons at the top of our website pages. When you launch the cell line browser, a list of all our current selected cell lines and their associated data is displayed. Selected lines are lines that have passed quality control and will be included in HipSci's final public iPSC resource. Many of these selected cell lines are available already to researchers from the cell line banks we deposit our lines with. If they are not available in the cell banks yet, then they will be soon. It is unlikely you will ever need to see cell lines which are not selected for banking. If you ever do, you can uncheck this box called Display Only Lines that are selected for banking. And you can now view all of the cell lines from HipSci's pipeline. This includes non-selected lines, lines in progress, and lines which failed QC. The default cell line browser displays a wealth of information on the lines. Each row is a cell line and each column gives you unique information about that line. So let's go through this. The first thing you will see is the cell line name. Each of our cell lines has a unique name. This standardized naming convention was proposed by Luang et al. In 2011 and was employed by HipSci. So let's use PodX1 as an example cell line to see what information is contained in a cell line name. These four letters HPSI represent an abbreviation for HipSci, so will always be the same. The following four digits represent the date the line was registered. So for example, this line was registered in November 2013. The letters following this tell you the cell type. An I stands for induced pluripotent stem cell. A PF stands for primary fibroblast, and ER stands for erythroblast. The following unique four-letter code is specific to a particular tissue sample from a donor. The final number denotes the iPSC line number from that donor. Multiple lines can be derived from one donor. If there is no number here, then it is either a fibroblast or an erythroblast. The next two columns are pretty straightforward. The disease status tells you which one of the 16 cohorts the cell line belongs to, and the sex of the donor from whom this particular cell line was derived. The culture column is particularly important for those who are purchasing our lines, as it tells you the culture conditions for that specific line. Note that we have cell lines that are both feeder-free and feeder-dependent. Adjacent to this is the purchase button, a direct link to purchase this particular cell line at the cell bank ECAC. So for PodX1, if we click purchase, we are taken directly to the ECAC page to purchase the PodX1 line. Next, we have the donor consent column, which categorizes all cell lines as either open access or managed access. It is really a reference to how any genomic and genetic data from these lines must be stored, handled, and distributed. I am going to explain in more detail about our managed access and open access cell line categories. The lines from our normal healthy donors fall under either managed access or open access, based on the ethics conditions they were collected under. All cell lines from donors with rare diseases will always fall under managed access. Managed access data is only available via a data access agreement. For the managed access cell lines, the recipient agrees that any genetic or genomic data it generates from the use of the cell lines will be held securely and only used in biomedical research. The data must only be made available to third-party researchers under a data access agreement at least as stringent as the data access agreements for data from the same cell line submitted to the EGA by Sanger. For open access cell lines, there are no data access restrictions, and the data can be freely downloaded. The recipient of the cell line agrees not to make any attempt to identify the original donors of the cell lines. They also agree to remove any data mapping to the Y chromosome before making genetic or genomic data available to third parties. Once this has been done, data may be made available for biomedical research in an open fashion, permitting redistribution of derived datasets. The next column refers to the bank status of the cell line. HipSci has established a banking arrangement with the European Collection of Authenticated Cell Cultures, ECAC, for distribution of cell lines to academic researchers, and the European Bank for Induced Pluripotent Stem Cells, or EBISC, for distribution to commercial entities. EBISC also distribute to academic researchers. The entire HipSci collection will be available from ECAC and a smaller subset from EBISC. The bank status column tells you where the cell line is with respect to it being available at these cell banks. So a capital B means the cell line either is or will be available from these banks. The colour of the B provides some more specific information. A red letter B tells you cell lines have passed quality control standards and will continue in the pipeline towards being banked at EBISC and or ECAC. A blue letter B tells you the cell line is currently available at ECAC. A green letter B tells you the cell line is currently available both at ECAC and at EBISC. A letter P stands for pending selection, and this means the cell line has yet to undergo quality control checks. And a letter N means the line has not been selected. These cell lines will never be available from a cell bank. Typically, this means another cell line from the same donor has been preferentially selected, or the line has failed to pass quality control checks. The final 9 columns represent the data that has been collected for each of the lines. The presence of a dot means the dataset is available. An in-depth review of the data is provided in the data overview video. Here, I will present a very quick overview of this data. Genotyping and expression arrays are used as part of our quality control checks on the cell lines we produce. We compare the original somatic cells to the derived iPSC lines to ensure genomic integrity has been maintained and iPSC cell lines are pluripotent. Cell lines which pass the quality control checks and are selected then progress to whole exome sequencing, RNA sequencing, and DNA methylation. Whole genome sequencing has been undertaken for a small subset of our open access lines and the somatic cells from which they were derived. Proteomics and cellular phenotyping assays are also undertaken on HipSci lines. The cell line browser allows you to easily filter for specific qualities you may be looking for in your cell line. Furthermore, you can choose to display additional information that is available under this More Columns button. You can further explore a particular cell line of interest by clicking on the line name. This will launch a new web page with specific details of the cell line you are interested in. So at the top here, you will see the cell type, and this can be either PBMC, fibroblast, or iPSC. The banking status links here will take you directly to the page where you can purchase the line itself. If you are using the lines for non-commercial research, then purchase the lines from ECAC via HipSci Collection. If you are intending to use them for commercial research, you can obtain the line from eBisc. Note: all lines available in the eBisc catalogue are also available to academic researchers via the HipSci ECAC collection, but for a lesser price. Next, we have the donor ID from which this particular iPSC has been derived, followed by the cohort the iPSC belongs to. The source material for the majority of lines is skin, but we have on the rare occasion derived cell lines from whole blood, so in these instances you will see whole blood written here. We have worked with numerous clinicians across the UK, and the tissue sample provider for this specific line will be stated here. Two methods of derivation have been used, so this can be either cytotune 1 or cytotune 2. Then we have the date the line was derived, followed by the age range, ethnicity, and sex of the donor, followed by the culture condition for the line, the biosample ID, and a certificate of analysis. In the COA, you can find the passage number and specific QC tests conducted on the line. So here, for example, you can see that the cell line has passed a number of tests, including mycoplasma, fluidyme, resuscitation, morphology, pluripotency, and Sendai clearance. Cell lines that fail any of the first 5 tests are generally not banked. However, a very small subset of lines fail to clear the Sendai virus, but are still a valuable scientific resource. In this instance, the cell line is banked, but the Sendai status is flagged to the end user at purchasing. We also provide a recommended standard operating protocol for all HipSci cell lines. Scrolling down the rest of the page, you will see all the QC and assay data files for download, followed by a link to the Encyclopedia of Protein Dynamics if you are interested in the proteome data. Last but not least, we have a graphic presentation of the quality control data for the expression array and the genotyping array. Okay, if you are someone who doesn't have a specific criteria in mind and are just looking for a cell line that is essentially a good clean line, we have put together a recommended panel pack that you will find at the top of the cell line browser. There are 2 panels here, a feeder-free panel and a feeder-dependent panel. The lines in both of these panels have been selected because they have high pluritest scores, low or no copy number variation, comprehensively characterized datasets, and a proven ability to differentiate well. This is now the end of the video. If you have any questions or queries about cell line availability, ordering, or purchased lines, you can email culturecollections.technical@phe.gov.uk. For any general inquiries, please email hipsci@ebi.ac.uk.